Good afternoon, everyone. Thanks the organizer for having me here. Today, I would like to talk about imaging the effect of hyperosmolality on individual tricellular junctions. I would first like to talk about tissue barriers. So they are the interfaces between the organs and the blood. Some important examples are blood-brain barrier and blood-kidney barrier. So usually these barriers consist of cell layers separating the blood and the organ, which keeps the organ from harmful substance in the blood. So there are mainly two pathways for transport across tissue barriers. One is transcellular pathways across cell bodies through ion channels or membrane receptors. The other one is paracellular pathways across cell-cell junctions, which is regulated by protein complexes called tight junctions. So today, the project I introduced will be mainly focused on studying the transport properties of tight junctions. So talking about tight junctions, here shows a hexagonal map of a cell monolayer. Based on the morphology, we can classify the tight junctions into two types. One is bicellular tight junctions where two cells meet, and the other one is tricellular tight junctions where three or more cells meet. So both types of tight junction, they are very small in scales, usually in nanometer scale or even in sub-nanometer scale, especially for tricellular tight junctions. So they are very hard to be studied by traditional techniques such as transepithelial electrical resistance technique. So to address these limitations, we use a technique called scanning ion conductance microscopy, SICM. So in SICM, we utilize a nanopipette, which is back inserted with a silver chloride electrode with reference to another silver electrode immersed in the bath electrolyte. So when this nanopipette is biased, it will generate an ion current when it is also immersed in the bath electrolyte solution. So when this nanopipette is brought very close to the sample surface, this ion current will drop greatly because of the obstruction of ion movement. So such ion current change can be depicted by the approach curve shown on the right. So with this approach curve, we can use it as the feedback to make the distance between the sample and the nanopipette constant when we raster scan the nanopipette over the sample surface. By doing this, we can obtain a topography map of the sample. So to further study the transport properties of tight junctions, our group later developed a more advanced mode of SICM, potentiometric SICM. We also call it PSICM. So in PSICM, we utilize a double barrel nanopipette with one barrel still used for basic SICM function and the other one for recording the local potential. So our group also modified the scanning protocol for PSICM. So during the scanning, when this nanopipette is brought very close to the sample for recording the topography height, it will pause for a fixed time. And at the same time, a working electrode beneath the sample will apply a transmembrane potential, which can trigger the response of the sample, which is further collected by the local potential probe. So after this process, the nanopipette will retract by a fixed distance delta z, and the same transmembrane potential will be applied to the sample, and the recording of the local potential will be repeated. So by doing this and utilizing the equation shown on the right, we can calculate the local conductance information of the measured position, which is related to its transport properties. So by this novel PSICM scanning protocol, we can obtain both topography map and the conductance maps of the cell sample simultaneously. So shown here is one example that I collected from a dog kidney cell line MDCK2 cells. So from both maps, we can easily identify the cell body regions, bicellular junction region, and the tricellular junction region. So today I'm going to talk about using PSICM to image the tight junction of MDCK2 cells under the treatment of mannitol, which is a hyperosmolar reagent commonly used for the treatment of brain injury. So shown here is a set of PSICM conductance maps collected under the different concentration of mannitol. On the top left is the control experiment where we did two consecutive PSI scans without changing anything. So we can see that there is no obvious change between two maps, which indicates that the tight junctions were stable during the scanning time. And when the mannitol was present in the system, we can see that the local conductance increased greatly at the tight junction areas. And more interestingly, this elevated conductance mainly occurred at the tricellular tight junction area instead of bicellular tight junction area. So to quantify this effect, we utilize computer vision algorithm which can help us extract the tight junction area automatically and then calculate the local conductance average at the different cell regions. So shown on the right is the quantified results. We can see that the tricellular junctions, its local conductance increased significantly after the treatment of mannitols, while the bicellular junction and the cell body did not. So this is very exciting for us because now we can differentiate the response of tricellular junctions and bicellular junctions to the hyperosmolar conditions with our PSICM technique. So next, we are going to study, we study the factors that may influence the tricellular junction opening under hyperosmolar condition. The first factor we studied is the concentration of calcium in a bath solution, which is non-necessary to the formation of tight junction. So in this experiment, I lowered the calcium concentration down to 2 micromolar, and we can see that the tricellular junctions opened greatly even when the mannitol concentration was as low as 25 millimolar. On the right column chart, we can see that under the same mannitol concentration, the tricellular junctions showed much higher local conductance change when the calcium concentration was low. So this can tell us that the lowering the calcium concentration can facilitate the tricellular junction opening under hyperosmolarity. So the second factor we study is the expression level of IL-1 protein in the cells, which is a non-tricellular junction specific protein. So in this experiment, we use transfection to have our MDCK2 cells overexpress ILDR1 protein as indicated by the immunolabeling and the immunoblotting results. And on the bottom left, we can see from the impedance spectra, the overall resistance of ILDR1 overexpressed cells is much higher than the wild-type cells, which indicates the successful sealing of tricellular junctions. So here shows the local conductance map, PSICM maps of ILDR1 cells under different mannitol concentrations. So from the conductance maps, we observed that actually we could not see the tight junction features anymore. So this tells us that the overexpression of ILDR1 not only sealed the tricellular junction, but also bicellular junction. And also after the addition of mannitol, we also did not see obvious change in the conductance map, which indicates that overexpression of ILDR1 can greatly reduce the opening of tricellular junction under hyperosmolarity. We also used immunolabeling to study the tricellular junction protein under the treatment of mannitol. So we studied 3 proteins ZO1 protein clotting to protein, which is specific to bicellular junction, and the tricellular protein, which is related to the tricellular junctions. We can see that the ZO1 and the clotting 2, they did not show obvious change. However, the tricellular junction protein tricellular, it shows delocalization under hyperosmolarity, which can explain what we saw from PSICM. To conclude my talk today, so we use PSICM technique to differentiate the response of tricellular junction and bicellular junction under the treatment of the mannitol. And we also see lowering the calcium concentration can facilitate this effect of hyperosmolarity, and this effect can be reduced by the overexpression of ILDR1 protein in the cells. And also, we can attribute this tricellular junction opening to the delocalization of tricellular junction protein and structure. So with that, I would like to thank my Baker Group member, the facilities in Indiana University, and Professor Jianghui Ho, my collaborator in this project, and the NIH funding. And thank you for your attention.